Hello everyone, and welcome to the Science North podcast, Give Vaccines a Shot, where we'll explore the latest vaccine news, learn all about the pandemic we are facing, and learn about the importance of our immune system. We'll be doing all of this through the lens of our experts. This month's guest is Dr. Gustavo Ybazeta. Dr. Ybazeta is a Bioinformatics and Genomics Associate at Health Sciences North Research Institute. His latest research has been studying wastewater in Northern Ontario to determine the level of COVID-19 virus in the general population. As Dr. Ybazeta will explain, wastewater analysis is not a new technique in research and has been used globally. It has been used locally and has allowed health units to determine the presence of COVID-19 virus in the general population. I was lucky enough to sit down with Dr. Ybazeta during an extremely busy time, and I'm happy to share our interview with you. And just as a little bit of an introductory, how long have you been working in the field of wastewater surveillance, and how did you come into this field of research?
I'm by formation, I'm an evolutionary scientist. I worked many years in genetics. My background is in genetics, and I've been working with genomics and bioinformatics. In the area of genomics, we were interested to obtain the genomes of different pathogens. And with that, we use the wastewater as a place where we can extract the pathogens and be able to sequence the whole genome of this bacteria. And that's why we start interested on sequence the virus of COVID-19 on wastewater. And that was happened. We started working with bacteria probably 5 years ago in wastewater, and now we are more than a year working with wastewater and COVID-19.
So when you said you were working with bacteria for about 5 years locally, you mean like in the Sudbury area, you were working with that?
Yes, we were working with one of the researchers here at the institute, and we were sequencing bacteria that we found it in wastewater, and some viruses too. To be more precise, prophages. They're viruses that they basically destroy bacteria. And there is a paper about them, like 13 new genomes that we sequenced.
Okay. Can you give us a sense of the history of using wastewater surveillance to monitor for disease?
Oh, it's a relatively long history. This is not new. If you want to think for idea to use the wastewater as a way to give you some information about epidemiology, we can start with one of the cholera epidemics in London in 1854. And for the researcher John Snow basically put together a map where he was able to trace the infections to a point of water that people they were going to collect their day-to-day water, and it was because it was contamination from the wastewater and the water that people they were drinking. And he was able to figure it out that based using the data, using from the patients, where the patients were happening, and he mapped that in the city of London, and he was able to identify the point of infection.
Okay.
We can call that was the beginning of the epidemiology and modern sanitation.
Right. So in that case, they were basically looking for the source of the infection or the source of where the pathogen was coming from.
Exactly.
But nowadays, when we do wastewater surveillance, like what exactly is being sampled? Like, are you sampling water from the sewage, from the plant?
Well, you can do many things. The thing that we're doing at this moment, we are going to the treatment plants, and before the wastewater is treated, after it was precipitated, we collect the influent with some solids, And in this case of the COVID-19, we're working with the solid part of that of that sample.
And so it's a measure of the virus in the environment, but it can't specifically tell you who's infected, right? So how do we use this from a public health perspective?
Well, the idea is that any product can be metabolite or infection that goes through our body and is deposited outside of us by secretion or excretion and ended in the pipes and concentrated in the treatment plant. We can measure it that. We can use different methodologies, like, for example, we can measure it metabolites produced by people that consume cannabis or opioids. But at the same time, any gastrointestinal bacteria or viruses, they go through our system and end it in the wastewater. The viruses, they are very interesting because they need to have a host in order to be replicated. The viruses, they are not
Right.
able to replicate by themselves, and if ended in the wastewater, that whatever is there, rules remnants, will give you an approximation of the number of people that they are infected, or the population that is infected.
And so would you be able to use it to measure things like how vaccination is working, whether or not it's reducing the amount of virus in the environment? Would you be able to use wastewater surveillance to do that?
Yes, we're precisely trying to get that information in order to make the comparison. One of the things that you obtain from the wastewater, you can see the changes of increasing or decreasing of the number of particles that we can detect using these technologies in the wastewater. And see these patterns of change, we can correlate it with measurements that we have done. Like, for example, if people, they are, proactively using masks, and we can see the decreasing of the number of particles, because it's no replication, it's no transmission. Anything that will be avoid the number of the virus to be replicated in organisms, there were no particles coming through the pipes, and we'll be able to measure it, that change after a few samples. um, yeah, that's the way that we can detect it, the success or not of the interventions, or even the levels of success of vaccination too.
So it doesn't just have to be vaccinations. It could be any sort of the non-pharmaceutical interventions that we've come through, as you mentioned, with the masks. That's really interesting. You had mentioned that you said that you were using the solid waste samples when you were collecting.
Yeah.
You were collecting solid.
Well, what we're collecting is a liquid portion, and in the liquid, obviously, there are some solids that they are in suspension. We centrifuge, and we concentrated that, and we think that higher percentage of the virus is in the solids. Probably some, they still in the liquid, but we are concentrated in the solids at this moment. Other groups, they are even trying to obtain information from the liquid part of that, but we have evidence that in the solid part you can find more particles.
And so are your samples kept sort of, I guess you probably don't exhaust all of your samples. Like, are you able to go back and sample retroactively if you're looking for specific things?
Yeah, well, one of the powers of the sampling is that you can sample and you can store the samples, and that's what exactly happened. They were able to retrieve a sample from previous times, and they were able to detect it. Another way is that when they're doing metagenomics, for example, when you sequence everything that is in a sample, sometimes people, they were not looking for some viruses, and they were able to, using bioinformatic tools to retrieve some of the evidence of the presence of the virus on those analyses that previously they were not being identified. Yes, it's possible to go and if the sample has been collected and was kept it in 4 degrees, it's possible to retrieve information even after long periods of time. Obviously, it will be a degradation eventually, but we can still recognize the signature of the virus in samples that have been collected, sometimes a month ago, but under certain conditions.
So the samples aren't kept like for years or.
We have evidence that even after 14 days, not only us but different labs have tested this. If you keep the sample 4 degrees, the normal temperature of a refrigerator, no freezing, because freezing seems to reduce the quality of the signature that we can retrieve from the sample. Um, when it's just cold, we can with no problem identify, after 14 days, and we stop doing the test because I think even probably can be longer than that.
Let's see. Oh, so another thing we often hear about is that you can use the wastewater surveillance sort of as an early warning system. I guess an example would be that sometimes you might detect the virus before somebody is sick. So how does that work?
Well, the way that it works is that anything that it goes through the wastewater, depending on the people that they can be, for example, asymptomatic, that number of particles will accumulate and concentrate on the when arrive to the treatment plant, and we can even detect, a number of asymptomatic people with these techniques. And sometimes, depending on the circumstances and the situations of each treatment plant or a geographical region, they can be able to detect from 3 to 4 weeks to just 2 to 3 days before you can detect before you have clinical patients in the hospital, because you have people that they are symptomatic and they are already shedding the virus in their feces in this case.
What is the method that you're using to identify the virus in the samples? Is it similar to a PCR testing?
It's similar. It's at the same time it's a bit different. Um, the As you know, the virus in this particular virus, it has a nucleic acid that is the RNA that is different than the DNA. The RNA, in order to be identified, we have first an amplification, and first we change it for RNA to DNA, and after that you have an amplification of that signature, and that cDNA or complementary DNA is what we measure it. We obtain normal. We target a specific gene that will be unique to the pathogen that you want to identify. In this case, in COVID-19, we target two different genes, and we obtain 200 to 250 base pairs. And through that, we can not only see the presence of that gene that by proxy indicate, a particle of the virus that it was in the wastewater. At the same time, we can quantify how many of those particles there are in the wastewater. And these two parts of the information, they are very important for us.
Who's been using the wastewater surveillance data
Where
in Canada or in
at this moment,
Ontario?
yeah, and this technology at this moment is using around the world in many countries in Europe. You have consortiums. In here in Ontario, we have a network, wastewater surveillance initiative. That is a total of 13 different universities and institutes, and they are even adding more people that they're helping us with that at this moment to increase a network. It's covering probably between 80 22% of the population of Ontario at this moment. And this information is used by the cities and the public health units of the areas where the samples are being taken.
And then when you were first monitoring the virus in the wastewater, were you comparing it to, like, we're not doing PCR testing sort of as widespread as we were before, but were you comparing it initially to the PCR testing that was happening?
Yes, before, we were able to probably until November, or I think it was the end of November, where the new rules for the clinical lab they changed because they were overwhelmed with the number of Omicron patients, and they were not able to test as before every single patient that will come to be or possible patient to be tested. Since then, those regulations they were changed. The wastewater basically became one of the few ways that we can detect the virus or the changes on the infection on the population. But before the time, we were comparing against hospitalizations and against the number of uh, assays, clinical tests that they were done, and they were very much the wastewater will be 2 or 3 days before you see those outbreaks or those increases in the clinical cases.
So they were tracking fairly well with the PCR testing before we didn't have that data.
Yeah, they were
Um,
basically mirroring one each other.
so was it? I guess this was sort of a way of being able to calibrate your data from the wastewater, or maybe what do you think was the more reliable data? Do you think the PCR testing was more reliable, or the wastewater numbers?
I don't think we can see one against each other, because they're different kinds of information that you obtain for them. Um, I would say they are more complementary because the clinical samples you are taking this directly for patients. The wastewater you're taking this from a wastewater. They are not. You don't need to have a contact directly with the patient or look for them. So this is a complementary data. I don't think one is better than the other one. Now you have some pros and cons, and the clinical data at the same time has some difficulties too. It all depends if everybody comes and be tested, or if you have the capability to test everybody. And it's, I think, more expensive if you go for every single person that is has some level of infection or have some symptoms. In the wastewater, you have just one sample that they can represent hundreds or even thousand or a million people in a specific catchment area that they will respond to your treatment plan. And with one sample, you can get that information. So I see more than one better than the other one. I will say that they are two different independent information about the same phenomena that you're trying to measure.
So where do you see the future of wastewater surveillance? Do you think it'll be important in detecting new variants, or like is it something that we're going to continue to do?
I think it's a big interest and has already proved that it can be very important not only for detection, for early detection, but even to monitoring how the development of the pandemic or outbreaks you have in your community. And this can be extended to other viruses or even to bacteria. But obviously, you need to modify the methodology and the kind of analysis depending of the conditions of the pathogens and the situations that you have for each treatment plan. Um, I think it will play a very important role in our communities, and now they are hyper connected. In less than a couple hours, you can be in a completely different continent. And this hyperconnectivity that we are living at this moment, not only physically, but through our computers, to everything, the level of knowledge that is moved to one place to other, and at the same time moving the people to one place to other, will need, some kind of systems that will be give us, early warnings, or even monitor, like we are doing at this moment, the development of these diseases that I think they're not only what COVID-19, but possibly will have another problems in the future, very similar or different of what we're having at this moment.
All right, so I think I'm gonna wrap this up, but before we leave, what is the least favorite thing about your job? What do you?
The least favorite thing, um, so I will say that this waiting for have the results. Sometimes we want the results even faster, and being able to see those patterns and move the information as faster as possible. Just waiting sometimes is a problem.
And
And
then what is your? Oh, sorry. What is your favorite thing about your job?
my favorite thing is that the breakage of the silos. Uh, in my career as a genomicist, I will be. If you tell me 2 years ago that we'll be talking with engineers, experts in flow and people designing drainages, I will laugh on your face. I'm learning a lot of how the water flows and how important is the rates of sedimentation in pipes for the kind of information that we want to extract from this, and the interaction with people. I have, if something good came from all the things, is the to be able to work with 13 universities, generously sharing their time, their efforts, their people, their knowledge, um, without expecting immediate recompense or anything like that. It has been an unbelievable experience. And I think that probably the best, the interactions with people at different levels, and the breakage of the silos. Now we're
Yeah.
working with the city and the public health unit, and it's an incredible experience.
I think the most interesting thing about wastewater surveillance is that it's an early predictor of the presence of a virus in the population. It can also be used to assess the effectiveness of measures that we're taking in the population to reduce infection rates, such as masking or vaccination. Thank you so much for listening. See you next month, where we'll explore more in the world of vaccine science. We also want to know if you have any questions. If you have questions about vaccines, viruses, or the immune system, or anything about this episode, please send us an email to vaccines@sciencenorth.ca. If you want to find out more, please visit our website, sciencenorth.ca, Give Vaccines a Shot, where you'll find explainer videos, school programming, and these podcasts. Also, follow us on Facebook, Instagram, Twitter, and TikTok. If you're listening or watching on YouTube, be sure to subscribe. Lastly, Give Vaccines a Shot is funded through financial contributions from the Public Health Agency of Canada. The views expressed herein do not necessarily represent the views of the Public Health Agency of Canada.